Hello, and welcome back to the Biology Domination Podcast, the show where we break down NEET biology in a way that actually sticks. I'm your host, and today is episode 23 of our 30-day Biology Domination series. Today's topic is sexual reproduction in flowering plants, a chapter that feels overwhelming at first, but is incredibly logical once you see the big picture. Whether you're cooking, commuting, or just lying down with your eyes closed, visualizing biology, this episode is designed to teach you the entire chapter through your ears alone. Let's dive in.
Let's start with the flower itself. A complete flower has 4 whorls. From outside inward, the calyx made of sepals for protection, the corolla made of petals for attraction, then the andrewsium, the male part, made of stamens, and finally the gynoecium, the female part, made of pistils or carpels. The stamen has 2 parts, a filament and an anther. The anther is where pollen is made. It's bilobed, with each lobe having 2 microsporangia, so 4 microsporangia per anther. The anther wall has 4 layers, epidermis on the outside, then endothecium, which helps the anther burst open to release pollen, then middle layers, and finally the tapetum. The tapetum is the innermost nutritive layer that feeds the developing pollen grains. This is called microsporogenesis. The microspore mother cells divide by meiosis to form tetrads of microspores, which mature into pollen grains. Each pollen grain is surrounded by a tough outer coat called exine, made of sporopollenin, the most resistant biological material in nature. It survives everything: acid, alkali, extreme heat. This is why pollen fossilizes so well. Now the female side. The pistil has 3 parts: stigma, style, and ovary. Inside the ovary are ovules. Each ovule has a stalk called the funicle, and where the funicle meets the ovule is the hilum. The ovule has protective layers called integuments, usually 2, with a small opening called the micropyle. Inside the integuments is the nutritive nucellus tissue. Inside the nucellus, the megaspore mother cell undergoes mitosis to produce 4 megaspores. 3 degenerate. Only 1, the functional megaspore, survives. This one cell undergoes 3 successive mitotic divisions to form an 8-nucleate structure, the female gametophyte, or embryo sac. Now visualize the structure. At the micropylar end, 1 egg cell and 2 synergids. The synergids have special projections called the filiform apparatus. They guide the pollen tube toward the egg. In the center is 1 large central cell with 2 polar nuclei. At the chilazal end, 3 antipodal cells. Count with me. 1 plus 2 plus 1 plus 3 equals 7 cells. But the central cell has 2 nuclei, so 8 nuclei total. 7 cells, 8 nuclei. Say it until it's muscle memory. Now pollination. Pollination is the transfer of pollen from anther to stigma. 3 types. Autogamy is within the same flower, geitonogamy is between different flowers on the same plant, and xenogamy is between flowers on different plants. This is true cross-pollination and promotes genetic diversity. After pollination, the pollen grain germinates on the stigma. The pollen tube grows down through the style and enters the ovule, usually through the micropyle. This is called porogamy, and it's the most common entry method. Once inside, the pollen tube releases its contents, 2 male gametes. Now, the most important concept of this chapter: double fertilization. The first male gamete fuses with the egg cell. This is called syngamy. The product is the zygote, with 2 N chromosomes. The second male gamete fuses with the 2 polar nuclei in the central cell. This is called triple fusion. The product is the primary endosperm nucleus, with 3 N chromosomes. These 2 events happening together is what we call double fertilization. It is unique to angiosperms. No other plant group does this. It was first observed by a Russian botanist named Nowoshin in 1898. You will see this in NEET. Guaranteed. After double fertilization, development begins. The zygote divides to eventually form the embryo. The primary endosperm nucleus divides to form the endosperm, the food store that nourishes the growing embryo. Endosperm develops in 3 ways. Nuclear type, where the nucleus divides many times before wall formation, is the most common. Cellular type forms walls immediately. Helobial is intermediate and found in monocots. Here's your coconut fact. The liquid inside a tender coconut, that's nuclear endosperm. The solid white flesh is cellular endosperm. Same coconut, 2 types. In dicot embryos, you have 2 cotyledons flanking the plumule above and radicle below, with epicotyl and hypocotyl regions. In monocot embryos, like wheat and maize, there is 1 cotyledon, called the scutellum, a sheath protecting the shoot tip, called the coleoptile, and one protecting the root, called the coleorhiza. After all this, the ovule becomes a seed, and the ovary becomes a fruit. And finally, 2 special concepts. Apple mixes is the production of seeds without fertilization, seen in citrus and mango, commercially very important for producing uniform crops. Polyembryony is when a single seed contains multiple embryos, also seen in citrus. Let's wrap up with your rapid-fire revision. Tapetum nourishes developing pollen grains. Sporopollenin is the most resistant biological material. The embryo sac has 7 cells and 8 nuclei, because the central cell has 2 polar nuclei. Double fertilization involves syngamy forming the zygote and triple fusion forming the primary endosperm nucleus, unique to angiosperms, discovered by Nowoshin in 1898. Coconut water is nuclear endosperm. Ovule becomes seed, ovary becomes fruit. Apomixis produces seeds without fertilization. That is the complete chapter in one podcast episode. Tomorrow we hit revision day 24, covering human physiology 2 and botany reproduction. Follow Hidden Wisdom Academy Biology, subscribe on all platforms, and share this episode with every NEET aspirant you know. Until tomorrow, keep dominating.